and welcome to our webinar. This is the 2025 SUDC Registry and Research Collaborative webinar on our lessons learned and future opportunities. I'll just start with introducing myself and then my co-presenter, Dr. Devinsky, and then we will get started, and I will also introduce our other study team members that are with us today, Dr. Leitner and Dr. Pinneri. So my name is Laura Gould. I'm a research assistant professor at NYU Grossman School of Medicine. And together with Dr. Devinsky, we're the co-founders and co-principal investigators of the registry here at NYU Langone Health. I have been studying in this field of SUDC research for, I can't believe, more than 25 years now. And my passion stems from a personal experience, unfortunately, when I lost my daughter, Maria, when she was 15 months back in 1997. And with all of the difficulties of loss itself, the added trauma of not having any answers around it really made it excruciating. And then on top of it, to find out that there was really no focused research on it, just made it all the worst. Back then, there wasn't a terminology like SUDC to kind of corral around or connect with. And so back then, I started first working with Dr. Henry Krause in San Diego. We started the San Diego SUDC project in 2001, where we first named sudden unexplained death in childhood and published some of the first articles doing a retrospective review of some children. And then in 2014, I was invited to join NYU Langone Health by Dr. Devinsky to propel forward our understanding of SUDC and to help families dealing with these tragedies. So I'd like to introduce Dr. Devinsky now as well for those of you who don't know him. But Dr. Devinsky is a professor of neurology, neuroscience, neurosurgery, and psychiatry at the NYU Grossman School of Medicine, where he has directed the Epilepsy Center for more than 30 years. His research has contributed to the FDA approval of more than 10 medications, including his role as the lead investigator for the only approval of a cannabis-based medication, for which Time magazine named him one of the top 10 people who transformed medical therapies in 2018. He's published more than 650 peer-reviewed papers, served on more than 20 board of directors and scientific boards for nonprofits, as well as companies focused on advancing novel therapies for disorders with high unmet needs. His academic work has consistently been cited among the top 1% of all American medical researchers. And personally, I want to thank Dr. Devinsky for all of your dedication to this. The collaborative, as I mentioned, was created here at NYU in 2014, and our aims have been very clear: to understand why these tragedies occur, sudden unexplained deaths in children, and to identify children at risk so that we can develop prevention strategies and keep them safe. NYU School of Medicine is based in New York City, for those of you not from the New York area. And although NYU has over 300 locations, we are based around the main campus, which is in the east side of Manhattan. And for landmarks, we're a couple avenues east of the Empire State Building. I would like to just stop a moment and say that I know that there are many affected families attending this webinar today. I would like to recognize you and to please extend and accept our deepest sympathies on behalf of our study team. I'd also really like to express our gratitude to the families who have enrolled in this research. It is really totally due to your courage and trust in us to learn from your children and your life's worst tragedy to be able to share any of this progress today. So thank you very much for that. Here's a slide that shows you a lot of the collaborators on the registry. You'll see many folks here at NYU Langone, many different clinical specialist researchers. You'll see some of the NYU core facilities that work with us on some of the experiments, as well as our external collaborators at different institutions. And off to the right, you'll see the many cardiac, neurological, forensic pathologists, and also a couple pediatric pathologists included in that. Next, I'd like to recognize two of our team members who are with us today, joining us to help us with this webinar. And that is Dr. Leitner, Dr. Dominique Leitner, and Dr. Kathryn Pinneri. So if you don't know them, I'm going to introduce them also with a little bit of a bio so you know their background. They are going to be manning the Q&A. So if you do have questions as we go through the presentation, feel free to jot them in the Q&A. And they will respond and address them. And at the end, we'll have some time also after Dr. Devinsky do our presentations to go over some questions and answers live as well. But Dr. Leitner is a neuroscientist with research interests in mechanisms of disease, particularly in SUDC, epilepsy, and neurodegenerative diseases. She began gaining research experience as an undergraduate student at the University of Arizona in the fields of biochemistry and neuroscience. This was followed by additional research training at the National Institutes of Health, and then a PhD at the Pennsylvania State University in neuroscience. She completed her postdoctoral training at Mount Sinai here in New York City, and that was related to stem cell biology. She's now a senior research scientist here at NYU, investigating mechanisms of disease using omics approaches, which you'll hear more about in a little bit, and analyses of human brain tissue to further understand disease processes. Dr. Pinneri is a board-certified forensic pathologist for over 20 years. She's currently the Assistant Chief Medical Examiner at the Office of the Chief Medical Examiner in Roanoke, Virginia. Prior to this, she was the Chief Medical Examiner of Montgomery County, Texas, and before that, she worked at the Harris County Institute of Forensic Science as the Assistant Deputy Chief Medical Examiner. She's been a member of the Disaster Mortuary Operations Response Team, known as DMORT, for over 20 years, and has deployed to mass fatality events such as the World Trade Center, the Haiti earthquake, and Hurricane Maria in Puerto Rico. She's on the editorial board of several forensic journals, serves on multiple committees for local and national organizations. She's the past president of the National Association of Medical Examiners and has been a dedicated study team member on the SUDC Registry and Research Collaborative since its inception. So thank you both, Dr. Leitner and Dr. Pinneri, for helping us today get through this and answer as many questions as we can. So our agenda here today is, I'd first like to go over some research concepts and definitions, just so everyone understands some of the terminology. I know everyone comes with different backgrounds to this. And then we will talk about our approach here at the registry, some progress, some highlights of our progress, and then our plans going forward. So the first one I'd like to talk about is this figure, which is the hierarchy of evidence pyramid. And all kinds of research is based on different types of study designs. And what you see in this triangle pyramid is that at the bottom, you have a lot of different papers across certain study designs. And the higher you go on the pyramid reflects the quality of the information or the evidence that you can gain from those studies. And so when we are looking at the registry and the case reviews we do, you'll see that on the case series and case reports. And with those types of studies, we can identify associations, but we cannot determine causation from them. We really need to work on climbing up that pyramid to study designs that can help us improve that evidence to further understand and achieve our aims. In terms of sudden unexplained death in childhood and the definition, this is the sudden death of a child, 1 to 18 years, that's unexplained after a thorough investigation. And this needs to be a comprehensive investigation, one that includes review of the clinical history, circumstances of death, the complete autopsy, and ancillary testing. Those are the additional tests that the pathologist would order based on what is clinically indicated to better understand that death. Another word you'll hear is a febrile seizure, and this is defined as a convulsion in a child which is provoked by a fever or illness, and it typically occurs between children 6 months and 5 years of age, And they're pretty common. It's the most common type of convulsive event in pediatrics. In the US, about 3 to 4% of all children will experience a febrile seizure. And in other parts of the world, like India and Japan, it's even higher, closer to 10%. They are considered largely benign, and they're not epilepsy. And one of the reasons is they're not epilepsy is for epilepsy, those seizures need to be unprovoked. And so epilepsy is defined as 2 or more unprovoked seizures or 1 unprovoked seizure, but where you have evidence on EEG or MRI consistent with focal epilepsy. About 3.5 million people in the US have been diagnosed with epilepsy. And when we look at the underlying causes of epilepsy, the majority of them, about 2/3, we don't understand why a person developed epilepsy, but we do know there's genetic markers in some, there's vascular, inflammatory, tumor, or if you have a traumatic brain injury, that can also increase your chances of developing epilepsy. Another word that you'll hear about is sudden unexpected death in epilepsy, or it's referred to as SUDEP. This is death in someone with epilepsy, but where any other cause cannot be identified from that investigation. Most research shows that SUDEP deaths are most common when the person's seizures are not controlled, and they tend to be nocturnal convulsions where the person is sleeping alone. So some of the risk reduction around SUDEP has been centered around really getting on medications that control the person's seizures well, or surgery to eliminate seizures. Improved lifestyle, adherence to medications, sleep hygiene, avoiding alcohol, and sleep supervision to help keep that person safe. 2 more definitions: sudden cardiac death, and this is defined in most literature as sudden cardiac death is due to a cardiovascular or an unidentifiable cause occurring within an hour of a symptom. And so I think most people, because they're like, wow, this kind of sounds similar to an unexplained death in a child, you don't have an identifiable cause. And that's true. And that's one of the reasons why surveillance around sudden unexplained deaths in children can be complicated. We also have terminology associated with unexplained deaths in infants, and there's many of them: sudden unexplained infant death, SIDS, SUDI, there's many. But when we look at these overall and we just step back and we say unexplained deaths in the young, we see that these things tend to kind of overlap, and the variability among the way they may certify might be variable. So we know that deaths due to epilepsy or sudden cardiac death can occur at any age, whether they're infant, a child, or an adult. Unexplained deaths in infancy, by definition, only occur in infancy, and unexplained deaths in children, again, only occur in childhood. But when we look at the surveillance that we do know, and this is statistics by the CDC of looking at those ill-defined causes, in 2023, there were 431 total deaths that were unexplained between the ages of 1 and 18, but 60% of them were among toddlers, ages 1 to 4. And you'll see that that directly overlaps with the peak of where the majority of febrile seizures occur. And so just keep that in mind as we talk a little bit more. In addition, sudden unexplained death in childhood is not a diagnosis. I think it's really important to reflect on that this is really a category of deaths that elude our scientific understanding. And as soon as we can explain a sudden death, that becomes explained, and we make the SUDC category hopefully smaller over time through successful research. But underlying with this is that we know there could be neurological mechanisms, cardiac causes, metabolic, genetic, infectious, and other things that we haven't considered. So that makes research even more complicated. We're not studying one thing. We're studying all the possibilities of what can cause such a sudden traumatic event. And more recently, we're seeing a bit of a trend and change in the way people are thinking about these seemingly related circumstantial deaths. And we're seeing people think of them more as kind of a continuum and looking at common factors associated. So here is a recent publication just this year and showing this Venn diagram of sudden unexplained infant death, SUDC, and SUDEP. And we know a lot of them are associated with sleep. A lot of them are associated with being found in the prone position or a slight association higher in boys. We also know that there's some genetic variants associated with sudden death. So this is really important to keep in mind that as we learn more, we're improving the research of surrounding related deaths as well. But one of the other things that we deal with in terms of trying to research SUDC is the barriers we face. One is that the U.S. medical examiner death investigation system is varied from place to place in terms of the resources specific offices have and the extent of those investigations, as well as variations in the way they certify a death, which will directly impact those numbers that the CDC reports. Different wording on the death certificate may not include those. Another concept to keep in mind is this idea of death certification, of what is put on the death certificate versus our medical understanding. And so when the pathologist identifies a structural or an anatomic finding, being able to identify a cause of death, there's going to probably be more consistency in those determinations from system to system. But when we have things that are functional or physiologic, things that are electrical, like seizures or heart arrhythmias, those don't leave evidence behind that are obvious from the autopsy based on standard testing that is done. And that is the other reason that we look at molecular mechanisms and different omic research to try to identify evidence that may be left behind that can help identify if any of these things may have occurred. So we've mentioned omics a few times. I just want to define it for you, that omics is the study of the biological processes in our cell functions. So we're all born with our unique DNA that makes us the unique person we are. And that DNA goes through a process in the way it functions in the body in terms of communicating through RNA, amino acids, and proteins that create the function of what that biological function means. So we're going to talk about some of the genetics that we've done in terms of the DNA. We're going to talk about some work that's upcoming regarding RNA. And we've also done some work in regards to proteins and proteomics that we'll discuss. And I hope that you can imagine that along this process, we can learn things at each stage of this process, and we might also be able to identify different changes that may be contributing to a disease process. So our approach at the registry, and for families who have enrolled, they know this very well. When they first enroll, we work with the family, we work with the medical examiner, coroner, to collect as much information as we can about that child. We collect all of this information, we review it, and then it goes through a very detailed review process that includes a lot of different types of specialists. And it's called a blinded review because everyone who reviews this does not know the original opinion of the original pathologist. They don't know the name of the child or where they're from. And this improves the quality of our research by decreasing any potential bias along the way. And so we think it's a very important process that we do. At the end of this, we return all of those results to the family, as well as the medical examiner and coroner. And so those child death investigations and case reviews that you see kind of in the middle teach us a tremendous amount. And we are studying close to 400 children at this time. And what we learn from them is also hypothesis generating. It makes us think of new research questions based on some of the things we learned from them. And those are some of the experiments that you see in those outside bubbles. And so what I'd like to do now is turn it over to Dr. Devinsky to really talk about some of our highlights in some of those areas.
Thanks so much, Laura. And this slide points out that SUDCRRC is really a collaboration between three primary groups. There are the families who really drive everything and have unfortunately suffered the most. There is the medical death investigation community, which we are closely affiliated with, and Laura, Kathryn have been part of NAME. I have gone to many of the meetings. And then there are researchers, including Dominique Leitner, who's with us today, and probably dozens of others who have participated in this process for the SUDC Registry and Research Collaborative. Next slide. And the next one highlights where we started. So this effort that Laura and I teamed up on back in 2014 gave rise to a set of publications which have continued. And I would say that, both proudly and sadly, I think the publications that Laura and I collectively have helped initiate and publish form the bulk, more than 50% of the literature on this topic, which I think is in some ways a success for us, but I would say is also a failure of the broader research community. This remains an incredibly underfunded area for the Centers for Disease Control, for the National Institutes of Health, and for other European and international research agencies and public health agencies. There are many examples, but SUDC claims many more lives than fire. Although if you asked, or SIDS, which is about 3-4-fold larger as far as the numbers, it would be hard to find a parent in the United States who hasn't personally learned about fire safety and who has not learned about SIDS. But with regards to SUDC, it would be uncommon to find anybody who's heard about it unless it impacted someone very, very close to them. Next slide. So this again looks at some of the clinical features in these patients that relate to what we think is susceptibility to having a sudden unexplained death in childhood. So as Laura pointed out earlier, the vast majority are toddlers. The median age is actually 22 months. For our registry, the majority of individuals are white and probably reflects a bias towards a slightly higher socioeconomic income, access to the internet, etc. But other things we see, I think, are biologically very relevant. Males are more predisposed, which is also true of SIDS and SUDEP and epilepsy kids. Sleep is when this occurs during apparent sleep in more than 85% of cases. Again, very similar to SIDS and also very similar to SUDEP, which is more like 60 or 70%. A history of febrile seizures is in our series close to 30%, in other series gets up to 33%. And I think it clearly, in my mind, speaks to, as I'll get to with the video analysis, that some of these children probably have febrile seizures and unfortunately die from them, which we know people without SUDC-related deaths have single seizures. And often if they occur in sleep, and often if the individual is not monitored, that is, for example, if it's an adult, they sleep alone, or if they're a child, there's no sound monitor or video monitor to alarm the parents, that those are going to be the individuals at greatest risk. Recent infection or illness occurs in more than 50%, and this again may tie into the febrile seizure history because many febrile seizures occur in children who are ill but don't mount a significant fever, and almost 50% of these children are face down, and that probably is just an unfortunate circumstance of either the way they sleep or where they land at the end of a seizure that puts them at greater risk of respiratory compromise. Next slide. So I'm not going to go through this slide in detail. We'll make the slides available, but we literally look at more than 1,500 elements from the child's medical records, family interviews, the medical death investigation, autopsy, pathology, and then our own blinded forensic pathology review. So there's an enormous amount of information that's captured in our registry and can be used for comparing, for example, neuropathology findings with clinical findings or anything, and will hopefully be a foundation for future research, not only for members of our collaborative, but beyond. Next slide. So here are some additional studies we've undertaken to increase our understanding of what causes SUDC. So here is an important paper, one that I find fascinating and was very informative. And this looked at the concordance between original death certifications and our panel. And indeed, you can see that there are many intrinsic and extrinsic causes of death. Intrinsic, for example, any cardiac abnormalities, pulmonary, neurologic, infectious. And those are present in a fair number of these cases. For extrinsic, and this is really the way that medical death investigation forensic pathologists divide things, the face down prone position is probably one of the more important ones. Next slide. And this is one that we have contributed a lot to. There is controversy in the literature. There, in our temporal lobes on either side of above our ears, is a part of our brain that's very important for memory. It's also a very common area for seizures to begin in, and likely impacted in febrile seizures. And the deep part of this hippocampus, it's a part of the brain present in rodents and many other animals, and critical for short-term memory and also related to some features of emotion. It turns out if you generate a seizure in a mouse, just put an electrode and stimulate it to cause a seizure, you can cause cells in the hippocampus to disperse. And it's a fascinating phenomenon. So if you look at kids who have had febrile seizures and die, they have more abnormalities in this area. And you can see some of the abnormalities. C is what I referred to is dentate gyrus dispersion. You can see those darker cells in the middle, just below the middle there. There's a sequence of them. They should really be about 50% as thick. They should be really highly concentrated in a thin line. If you look to the right on D, you see a very thin line, but it also reflects some thinning, as does B above. So there are many different types of abnormalities that can occur. The big million-dollar question is, are these specific to SUDC or seizures, or could they be what we call an epiphenomenon, that they're often associated, but they're neither the cause nor the consequence? I do believe seizures from experimental studies in animals can cause these, even electrical discharges that are far subclinical. I could be having those right now. You could be, and I wouldn't know it myself. You wouldn't recognize it. They're essentially non-detectable if you don't have an electrode stuck deep into the brain. That even those electrical discharges that don't cause any symptoms or observable signs can lead to this type of dispersion. So it's hard to know. So what we did is we did blinded reviews when pathologists go in with a hypothesis saying, I think these dentate gyrus or hippocampal abnormalities are the cause. Human bias is very powerful, and Dominique led this study. And what we did is we looked at these hippocampal findings in controls, in cases without febrile seizures, in cases with febrile seizures, and board-certified neuropathologists reviewed the slides but had no idea which of those three categories they came from. And the bottom line, as you see on the left, yes, it's not uncommon to find these in our SUDC cases, but essentially the febrile seizure cases are no different than those without febrile seizures. And controls, for example, a child who is otherwise healthy and died in a car accident, would also have similar numbers if you didn't tell the pathologist what they're looking for. Humans, as we all know, are incredibly biased, and scientists and doctors are no less so. So we have to really blind ourselves to try to get close to the truth. Next slide. Another category of studies that we've done, and this is in the omic category that Laura referred to earlier, are looking at different genetic biomolecular features. And this specifically looks at proteins. Again, just remember, you have a gene that's DNA. It is transcribed into RNA, which is then sent from the nucleus to the cytoplasm, where ribosomes will translate the mRNA into proteins, and the proteins do a lot of the work and structural jobs that are required in our cells and in our bodies. So these are cases where we looked at both the hippocampus and the brainstem, comparing those initially with SUDC versus explained deaths, and those with SUDC with or without febrile seizures. And we did find differences in all of those. For the SUDC cases versus children who died from, let's say, a car accident, we found many changes in protein levels, mostly related to inflammation and cell function. In the brainstem, we found abnormalities in the medulla, the very base of our brainstem that controls respiration, including serotonin receptors, which are vitally critical in regulating the breathing cycle. And we know young children are sensitive to this, especially during sleep. And then in those without febrile seizures, we also found abnormalities in inflammatory protein pathways. And all of these speak to the fact that these children likely have some baseline level of dysfunction in some metabolic protein pathways that do make them more susceptible to SUDC, although we can never be sure how much of these findings are a result of whatever led to the SUDC, including a lack of oxygen going to the brain, which is probably common in almost all of these children, sadly, before they die. Next slide. So what do we know about the association of SUDC and seizures? As I said, febrile seizures are present in give or take 3% of the general US pediatric population. They're present in close to 30% of SUDC victims. Keep in mind, again, SUDC median age is 22 months. The range for febrile seizures is 6 months to 6 years. So most of these children are dying before they would actually manifest febrile seizures. So if you did a cutoff of 22 months for the average US child, the febrile seizure numbers would be more like 2%. And it's likely that we believe, and you'll see some evidence to support that in a minute, that many of the SUDC cases probably had febrile seizures, but they were never observed or recognized by the parents before death, or indeed the very first one the child has was sadly fatal. Sleeping is a risk factor, especially if the child is in the prone face-down position, which can obviously impair breathing. We have death recordings that we'll review in a minute, hippocampal anomalies that I referred to before, which do not remain proven, but highly suspicious. And then finally, genetic findings that we'll also cover in a few minutes. Next slide. So this is a review article that Laura and I participated in, and it was asking the question, are febrile seizures benign? And there are two ways to look at febrile seizures. If you go to the average pediatrician, and keep in mind 3% of the kids that pediatrician cares for is febrile seizures, and say, are they benign? They'd say, oh, absolutely. I reassure the parents it'll be outgrown by age 5 or 6, and the kids are usually fine. And that's usually true. Yes, a much higher number of them do go on to have epilepsy, maybe 5-6%, as opposed to 2% of the pediatric population. But they do have an increased risk of mortality. Now, it's not common, and it's not something that any single pediatrician is likely to see. But if you take all the pediatricians in the state of New York or California or Illinois, there will sadly be many of these. And so with febrile seizures, we know that hippocampal histopathological abnormalities are common. And there are also frequently neurodevelopmental issues, which are relatively mild, can be attention deficit disorder, language comprehension disorder, irritability, things like that, that are not these hard things like they're weak on the right side of their body, but they are definitely abnormal when you compare them to control populations. Next slide. So this is a video analysis that Laura led and I participated in with others. And it's a tragic study in that we actually had video recordings of toddlers who died. These were often kids who had crib cams when they were infants to make sure there was no SIDS type event prevented. And then after the kid was at age 1, the parents left it in. And sadly, we were able to identify 5 such recordings of a child with SUDC in which the entire night before they died was recorded. Now, these videos were shown to, I think, something like 14 different physicians, medical examiners, neurologists, sleep physicians, and it was almost unanimous in all 5 cases that there was a convulsive event where the child had a sudden stiffening, then became flaccid, breathing stopped, and then they died. This was published in one of the leading neurology journals, Neurology. It was highlighted on NBC Nightly News and the Associated Press, received a lot of attention. We, of course, none of these kids had respiratory monitors, cardiac monitors on, so we couldn't exclude what came first and what happened. But I would say, this is very, very strong evidence that these kids, only one of whom had a febrile seizure identified in life, that 4 of them with no history of febrile seizures had what appears to be a convulsive seizure and then died afterwards. Whether they were febrile at the time, we don't know. The exact pathophysiology, what happened to their heart and their lungs, their breathing, we don't know, but it's essentially a smoking gun. We have an evidence of what the murder weapon was, and we think it was probably a seizure, tragically. And it may turn out that seizures are the cause of most sudden deaths in these children and potentially in infants as well. Next slide. So this is another case-controlled study of febrile seizures and sudden death. And it was an anonymous survey of cases, febrile seizure with SUDC versus febrile seizure cases in controls. The SUDC cases were younger. As I said, febrile seizures can go to age 6. And for the febrile seizure patients who did succumb to SUDC, they were more likely to have developmental concerns, and we're not sure what this means, less likely to be reported to drool and less likely to wake up a lot during sleep. So maybe had a decreased arousal threshold compared to the average kid, which may put them at risk. Next slide. And febrile seizures also occur, as I said, very commonly in the general pediatric population. And we are doing an ongoing prospective study with 2 of the companies, Nanit and SAMI, that have monitoring devices for young children, to start to record large numbers of these events and start to see what do febrile seizures look like in sleep. We really don't know. It's almost never been seen, very, very rarely recorded, and sometimes we have just the descriptions of parents who are obviously traumatized seeing their young baby seizing, and eyewitness descriptions in those cases can be misleading. So these recordings will be invaluable. Next slide. And then just to point out, I hinted at this before, but if you look at all children in the United States between ages 1 and 4, it's an enormous number, give or take 100,000. And then if you go down to those who have febrile seizures, you're looking at about 3% of that, or 3,000. And then if you look for SUDC and SUDC with febrile seizures, they become much, much smaller numbers. So in part, these are the real numbers. We're not trying to inflate them. We do think SUDC is significantly underdiagnosed, because if a medical examiner gets a 2-year-old or 3-year-old who died in their bed, it's not a satisfying answer when the parents say, what caused my child's death? For the medical examiner to say, I don't know, and the child, it's not SIDS. SIDS is a name everybody knows, or SUID, which is what we call it now more frequently. SUDC is essentially unknown to 99.9% of parents in this country. And so if the medical examiner says, oh, it was an SUDC, that's like no answer at all. So they literally, and I've spoken to parents who have said, the medical examiner said, I didn't want to put nothing, I don't know. So I put something. The kid had a mild bronchitis, which is probably true for 15% of American kids during the winter. It's not a cause of death. And so in those ways, SUDC is underdiagnosed. Next slide. And so in thinking about this, it is important, I think Laura, myself, Dominique, and Kathryn, we're strong advocates for the SUDC community, but we also have to balance it. We can't scream that every child between ages 1 and 4 should be monitored every night for SUDC, because as you can see, we'd be monitoring 100,000 kids every year to try to detect maybe 400 events, 500 events per year, which is a lot. But even if you have parents with cameras and alarms, we all know that people oversleep, people forget to recharge the battery. And so it's really hard to balance scaring 100,000 parents to maybe save 12 lives. I don't know what the numbers are. No one does. What we need to do is find those children who are at very high risk so we can tell those parents to monitor very closely. But we do have good ways. Some of these cameras can detect convulsive movements and alarm the parents in their bedroom, which may be 30 or 100 feet away from the child, and potentially be life-saving. Next slide. And then finally, finishing up with some of the genetic studies that the SUDC-RRC has been very involved with and has led. This is one that we did with colleagues at Columbia, where we looked at 124 SUDC cases, and we did whole exome sequencing. So if you're just some basic genetics, we have 3 billion DNA letters in our DNA. About 1.5% of those code for the 20 or 22,000 genes that we call genes that code for proteins. So the whole exome sequences that 1.5%, which is in medicine the most important 1.5%. It's not the only important one, but it's the most important. And we found that about 9% of the children that we sequenced, we did find pathogenic mutations, either in genes that affect the heart or the brain, with interestingly the most common denominator across those mutations being those that affect calcium channels, which are critically important for the excitability of both heart cells and brain cells. And in many cases, that can extend across both tissues. Some of these abnormalities may affect both the heart and the brain and may make these children much more susceptible. We also found de novo mutations, which means we see what's unique about our group is we sequenced not just the child, but we sequenced the parents. So if there is a variant present in the child that was not present in the parents, and it was in a gene known to be related to critical cardiac or brain functions, we could be pretty darn certain that that is a cause, because in general, any of us in our protein coding genes only have one or two new mutations that are not present in our parents. And so this is very powerful finding, and it's an OR or risk ratio of tenfold. So these non-synonymous de novo mutations, non-synonymous means it causes a change in the amino acid sequence of the protein. De novo means it's a new mutation. And the chance of finding that was tenfold greater, which is highly, highly significant. Never mind 0.05, you're talking 0.00001. Next slide. I think that may be it.
Yes.
Yes, so I'm going to turn it back.
Thank you so much, Dr. Devinsky. So I hope that that is helpful in terms of understanding just some of the highlights of the work that we have been doing. As you remember on that slide, there's a lot of bubbles, and we really kind of chose and picked through some of the highlights to share today. But we have a lot of priorities ahead. I would say these are probably our top 10. But all of the information that we learn leads to that next research question in terms of moving the needle to improve our understanding. We would like to reopen our registry enrollment. We have slowed it this year due to reduced funding. We have done some pilot studies looking at machine learning to identify risk factors, right? Besides trying to understand the underlying cause, we want to be able to identify risk, children at greater risk, to be able to help pediatricians better care of these children. And in terms of some of our initial machine learning, just using some of our data, not the 1,500, but some key factors, that seems a really promising lead. Artificial intelligence to look at video analyses, comparing those videos of the febrile seizures in living children during sleep versus some of the SUDC videos we have. Can we identify risk factors in them? Can we identify protective factors in them? Dr. Leitner and our group is also working with one of our cores here at NYU to look at image analysis, artificial intelligence, to better understand the normal architecture of the hippocampus. We've talked about that that is a complicated area of the brain where there's different opinions on that, and we can really use artificial intelligence to give us an unbiased review of that. Moving on from whole exome sequencing, whole genome sequencing, looking at that other part of the genetic material that we have to see what that can teach us. We just received a grant to do, Dr. Leitner received from SUDC UK to look at the transcriptome, so the RNA in SUDC, and that will be the first time that anyone has been able to do those types of studies. And that also lends itself to some really exciting areas of doing multi-omic studies, taking what we learned from the genes and our genetics and the proteins and also the RNA and looking at these different data that we have and combining them is a really, really strong avenue as well. We've also identified some novel genetic variants that have not been reported before. Functionally characterizing them in the lab and sharing that with the medical community will be important. And what can the microbiome teach us about SUDC in terms of risks as well as causation? And then those things that we learned in the case control study about sleep and sleep quality, arousal systems, also warrants sleep studies in this area. So, at the top of this, we said, priorities ahead as funding permits. Obviously, we have a large team working at the registry of just incredibly dedicated people. And we will be able to charge forward as fast as we can if we have the appropriate resources to do so. Some people have asked, how can we contribute? How can we support directly? And so there's really three ways in terms of financial support. We do have our registry website, which is at sudcrrc.org. It takes about 5 minutes to create a personal fundraising memorial page, if that's something that someone wants to do, or holding an event to benefit. Donations are all 100% tax deductible. NYU is a nonprofit organization. And if anyone has any questions about these, they can email us they'll show the email at the last. But this, funding is our biggest barrier at this point. We have all of the other critical elements, but funding is our biggest concern going forward. And with all of the highlights and advances, I think, as scientists, as families affected, as researchers, this is the time when we want to charge forward, speed things up, not slow down. We want to find these answers and start to prevent these deaths from occurring. So I'd like to thank again all of our team here at NYU and abroad, our sponsors, and again, thank you to the bereaved families. When we started this in 2014, I think there was a lot of concern that, you're not going to find families to participate in this. And I felt very differently. I said, no, families will be leading the charge. They want answers more than everyone, and learning from our child's tragedies to improve the future for other children is very important. So we will stop there. We will go through and answer a couple questions. And I'll stop sharing my screen, but I do want to just reiterate the email address here. Those come to me, but I can kind of triage to our study team members that will be able to help answer the question if it's not in my wheelhouse. And I will stop sharing my screen and open it up a little bit. I also wanted to mention, for people who want a deeper dive into some of these areas of research that we're doing, there is going to be a conference in October. It's called the ISPID conference, and it's all virtual. And virtually on Thursday of that conference, we'll be doing presentations all day on the details of a lot of the studies that we talked about. I think we have 10 different presentations between our researchers at NYU and our collaborators. So we're excited about that, and I will send out an email with information on that. Before we go to any of the Q&A, I'm just wondering if Dr. Pinneri, if you have any thoughts that you wanted to share, and then maybe go over to Dr. Leitner if there was anything you felt like we didn't cover or you thought you would add from your point of view.
Sure. I'm typing away at the wonderful questions in the chat or the Q&A. But first, I really just want to say how grateful I am to the families who choose to participate in this very important research. From a forensic pathologist standpoint, you have made me a better forensic pathologist. I have a better understanding of the depth and detail that these deaths require. And it's really actually made forensic pathologists all across the country better. Unfortunately, we are still limited in what we can and can't diagnose from the standpoint of the autopsy. And the testing that this registry collaborative is performs really is not possible in a standard medical examiner's office. We don't have those resources. We don't have the funding. We barely have funding to keep the staff necessary for our caseload. So it's a really, really important project. And, as more information becomes available, we'll have more answers, hopefully, for families, that experience the tragic loss that that many of you on this call did.
Thank you so much. Dr. Leitner, do you have anything else you might want to add?
Hey, no, that was a great overview. I'm happy to be a part of this group and have the opportunity to work on these studies. There have been a lot of questions coming in through the chat, some of which are related to the hippocampal neuropathology and omics. So those are more of the studies that I'm focusing on. And I think we briefly talked about that, but I'm also, I think you also have the publications listed on the website. So if people are interested, they can also look at those. And like you mentioned, there will be additional presentations in October at ISPID to detail some of that research further. We've had some of the first proteomic research studies on the brain tissue from these cases and have found some interesting results. So hopefully with additional studies, and more data, we'll be able to better understand the underlying mechanisms and identify potential biomarkers of risk.
No, thank you so much.